Assalamualaikum and good morning everyone. Okay, so today we are going to discuss about the classification of enzyme. So um, basically, okay, the classes of enzymes are oxidoreductase, transferase, hydrolase, isomerase, ligase, and Ligase. Okay. Now let's go one by one. So before that, this is my student. She came up with this mnemonic on how to remember the classes of enzymes. So she came out with over the hill. Over means oxidoreductase. The is for the transferase. H for the hydrolase. And I is the isomerase. L ligase, and the other L for the ligase. Now let's go to the first class of enzyme, which is oxidoreductase. Any enzyme that is in this class, okay, those enzymes catalyze the oxidation reduction reactions. Okay, when an oxidation reaction occurs, reduction reaction also will occurs concurrently. Okay, so what happens in this oxidation process or oxidation reaction? Oxygen is transferred from one molecule to another molecule, while in reduction reaction, what happens? Hydrogen or electron is transferred from one molecule to another molecule. Okay, so we take one example here. In oxidase reaction, meaning to say reaction that involve oxidation of a compound. Okay, so enzymes involved in oxidase reaction will catalyze the addition of oxygen to a molecule. So example, we have oxygen with glucose with oxygen. So we have gluconic acid and water. So meaning to say, right, oxidase catalyze the formation of gluconic acid and water from glucose and oxygen. And then dehydrogenase reaction, okay, where enzymes in this reaction, such as dehydrogenase, catalyze the removal of hydrogen or electron from a molecule. Okay, now easy to remember. The keyword here is DE. D. This means removal. Hydrogen, so means say the removal of hydrogen. So whenever you come across the dehydrogenase reaction, means say that reaction involves the removal of hydrogen or removal of electrons, right? And usually the enzymes here is known as dehydrogenase. So one example here is dehydrogenase catalyze the formation of oxaloacetate, NADH, and hydrogen ions from malate and NAD. Okay, next we have transferase. So meaning to say enzymes that catalyze the transfer of a functional group from one donor molecule to an acceptor molecule. And then this functional group can be methyl, acyl, amino, phosphates. Okay, we take three examples here. In phosphorylase reaction, okay, what happens? Phosphorylase catalyze the transfer of a phosphate group. So one example, in this within this phosphorylase reaction, glycogen phosphorylase catalyze the transfer of phosphate group into glycogen to form glucose 1-phosphate, or phosphorylase catalyze the formation of glucose 1-phosphate from glycogen and inorganic phosphates. PI means inorganic phosphates. Okay. Now in kinase reactions, what happens? Hexokinase catalyze the formation of glucose 6-phosphate and ADP from glucose and ATP. Okay. Then another example here is transaminase reaction, where this transaminase enzyme catalyze the transfer of amino groups from amino acid into a keto acid. Example here is transaminase catalyze the formation of alpha ketogluteric acid and alanine from glutamic acid and pyruvic acids. Now, to be more detailed, what happens? Transaminase, okay, transfer the amino group from glutamic acid into pyruvic acids. So we have alpha ketogluteric acid and alanine. So meaning to say this glutamic acid, when the amino group from this alpha glutamic acid is removed, this glutamic acid becomes the alpha ketogluteric acid. Then the amino group from the glutamic acid is shifted or transferred to the pyruvic acid, right? So the pyruvic acid, when this pyruvic acid receives the amino group from the glutamic acid, this pyruvic acid becomes alanine. Okay, this transaminase reaction is very important because this transaminase reaction synthesizes non-essential amino acids. Okay, and one example of non-essential amino acid is alanine. Okay, just imagine from glutamic acid we can get alanine, which is one example of non-essential amino acid. And this non-essential amino acid is needed by our body, right? If you recall, what's the difference between essential and non-essential amino acid? Essential amino acids, okay, those are amino acids that our body can synthesize, but non-essential amino acids are amino acids that our body cannot synthesize. Now, hydrolase, these are enzymes that catalyze the hydrolysis reaction. So in hydrolysis reaction, what happens? Complex substances are broken down into simpler substances by addition of water. Always remember that, okay, by addition of water. And hydrolase mostly are digestive enzymes such as pepsin, lipase, amylase, rennin, okay, trypsin. So these are three examples of enzymes that are grouped in the hydrolase. So maltase catalyzes the breakdown of maltose into glucose with the presence of water. Or maltase catalyzes the formation of glucose from maltose and water. Then lipase catalyzes the breakdown of lipase catalyzes the breakdown of triglyceride into glycerol and 3 fatty acids with the presence of water. And sucrase catalyzes the hydrolysis or breakdown of sucrose into fructose and glucose with the presence of water. Okay, next we have isomerase. Alright, so isomerases or isomerase are enzyme that catalyzes the conversion of an isomer to another isomer by rearrangement of atoms in the same molecule. So now these are examples of isomerase reactions. So we have phosphoglucomutase that catalyzes the isomerization of glucose 1-phosphate into glucose 6-phosphates. Another example is phosphohexoisomerase that catalyzes the isomerization of glucose 6-phosphate into fructose 6-phosphates. Alright, next we have ligase. Okay, so enzymes that catalyze reactions that joins two molecules with covalent bond into a process coupled to hydrolysis of ATP. Okay. Example here we have DNA ligase. So DNA ligase basically this enzyme, all right, combine or I should use the word ligase. Okay, so DNA ligase shorter DNA fragments into longer DNA fragments. Okay, Now, for this ligation of DNA to occurs, okay, ATP is needed, and when ATP is needed, obviously the ATP will be broken down into ADP with inorganic phosphates. Okay. Now, another enzyme that is grouped in the ligase is amino acid tRNA synthetase, which you already came across earlier. Okay, in chapter 2, if I recall. Okay, so amino acid tRNA synthetase catalyze the formation of glycine tRNA with AMP and pyrophosphate from glycine, tRNA, and ATP. Okay. And finally, we have Ligase, means enzymes that catalyze the breaking or formation of double bonds, and then in a molecule by means other than hydrolysis and oxidation. So now, example of reaction that example of enzymes, okay, that is grouped in the ligase is liases, okay, or ligase is decarboxylase and carboxylase. So decarboxylase, again we have the word here D, okay, carbo, so meaning to say the removal of carbon dioxide. So decarboxylase catalyzes the formation of acetyl coenzyme A with NADH, hydrogen ion and carbon dioxide from pyruvate, coenzyme A and NAD. Then carboxylase, or fixation of carbon dioxide. Okay, carboxylase catalyzes the formation of phosphoglyceric acid from ribulose B-phosphate, water and carbon dioxide. So that's it for classification of enzymes.